All right. Good morning, good afternoon, and good evening. Welcome to the first Biological Magnetic Resonance seminars. This seminar series is an effort by a number of us to fill the gap of the lack of biological NMR and EPR meetings this summer because of COVID-19. One of these meetings is ICMRBS, the International Council on Magnetic Resonance in Biological Systems, which has been postponed to July 2021. The scientific scope of ICMRBS spans solution NMR, solid state NMR, and EPR. So this seminar series aims to showcase similarly diverse and excellent talks by international speakers. We hope that these talks will inform all of us, especially graduate students and postdocs, about emerging topics in the field and inspire us to come up with new ideas so that when we go back to our labs, we'll be all the more ready to advance the frontiers of biological NMR and EPR spectroscopy. Our speakers for this series will include both well-established people in the field and up-and-coming young investigators. The speakers for the first 4 weeks have been contacted. For future talks, we welcome your suggestions. Please feel free to write to one of the 6 conference organizers. Nomination as well as self-nominations are both fine. Please send short emails, no more than 100 words, about the talk content and cite 1 or 2 papers on which the talks will be based. Some of these lectures will be posted or will be recorded with the permission of the speakers, and they will be posted on this ICMRBS YouTube channel shown here. We ask the audience to please abide by the honor code of not making private recording of any of these lectures. So this talk series will occur every Thursday so far, as we agreed on, at 10 a.m. Boston time, which is 7 a.m. California, 3 p.m. London, 4 p.m. Berlin and Paris, 5 p.m. Jerusalem, 10 p.m. Shanghai, 11 p.m. Tokyo, and midnight in Sydney. All right, so with this, I would like to transfer the virtual podium to Oscar Millet.
Hi, thank you, May. So this is Oscar Millet, and I am the organizer of EuroMar for this year, that it was supposed to happen in July, and it's been transferred until December due to the COVID-19 pandemic. As May very well said, this is an extraordinary situation that forces us to find new ways of communicating in the community, and this is a strategy, an idea that some of us consider that it would be beneficial for all of us. With that said, I will just very briefly introduce our first speaker, which is Luis Kay. Actually, Luis Kay doesn't need at all any introduction. I mean, he is absolutely well known in the community. He has made outstanding contributions to the field. He has received many awards.
I guess we then have to start. And I mean, there's this initial transparency. Please don't record. We anyway record and read all the rest yourself. So today's lectures will both be recorded and then put on the YouTube channel afterwards. And I think, Luis, you can start now.
Okay, so first of all, thank you very much for the opportunity to speak today and for that nice invisible introduction. I'm actually going to be talking about solution NMR, the big and the invisible, but today the big and the invisible are going to be a little bit different than typically what I speak about when I speak about these subject materials. So when I talk about NMR, the big, of course, I'm referring to supramolecular systems or molecular machines that have aggregate molecular weights on the order of many hundreds of kilodaltons, upwards of a megadalton. And over the past 15 or 20 years or so, we've really examined some of these machines in detail using C13H3 methyl labels, deuteration, and a methyltrosy. For example, we've looked at the proteasome, which gives very nice spectra. We've looked at a protein called P97, which is a protein involved in proteostasis, again, lovely spectra. We've looked at disaggregases such as ClipB. We've looked at other proteins that are involved in housekeeping, such as the ClipP protease. But what I want to talk to you a little bit about today is the nucleosome core particle, or referred to as NCP. This is a protein DNA complex. It's comprised of 4 different proteins. These are histones, and they come in pairs. So there's an octamer of histones, and surrounding the octamer, wrapped around it, is going to be about 150 base pairs of DNA. Over the years, we, in collaboration with Ye Wen Bai, have assigned the methyl groups of the histones. Shown here is the assignment, and we focus primarily on the proteinaceous components of the NCP. But of course, the NCP, the nucleosome core particle, is made up of DNA, and it's really the protein that serves the DNA, not vice versa. And so we'd like to be able to extend our studies to looking at DNA, and in particular large DNAs and DNAs that are associated with large complexes. So the question is how to go about doing that. Of course, one possibility is to remember that DNA has T nucleobases, and these nucleobases contain methyl groups. And so one might imagine just C13H3 labeling all of the T's in the DNA. The problem is that we want to look at very big DNAs, so there's going to be a lot of T's, and that's going to really seriously limit the resolution, even at the highest magnetic fields. And so what we would like to be able to do is to bring methyltroside to nucleic acids, and we're going to borrow a page from Mother Nature. Mother Nature recognizes the importance of methyl groups. It's designed methyltransferases, and in the context of bacteria, these methyltransferases come in pairs with restriction endonucleases and form a simple immune system, so that the host bacteria is going to be specifically acted on by these methyltransferases. The DNA is going to get methylated at specific places, and that means that the restriction endonucleases are not going to be able to cut up the DNA. The methyl groups protect the DNA from being endonucleased. By contrast, any foreign DNA that enters the cell would not have the methylation patterns and therefore would be cleaved. In the case of eukaryotic systems, methylation is also very important and is an epigenetic mark. It's important in the regulation of transcription in the context of methylation of DNA, and in translational regulation in the context of RNA. And methylation predominantly occurs, as indicated here. We have bases, so at the C5 position of cytosine, there's methyls, and also at the N6 position, there's a methyl in the case of adenine, and I'll be speaking about these two situations here today. So what we want to do is exploit Mother Nature, but we want to come up with a simple one-pot reaction scheme that is going to allow us to label our DNA of interest. And so what we do is we have the following situation indicated in panel A. We purchase C13H3 methionine, which is relatively inexpensive. To that, we add ATP and an enzyme that we produce called S-adenosyl methionine synthase. We also add the methyltransferase, or several of them, depending on how many methyl groups we want to add to our DNA, which we have to add in either the protonated or deuterated form. And then what happens is the methionine is going to react with the ATP to form S-adenosyl methionine, which is then able to transfer the methyl group to DNA with the help of the methyltransferase. And we can follow the reaction if we want by NMR. So this is a carbon-proton HSQC. At time 0, we have a very simple spectrum that consists just of the methyl group from methionine, which then evolves into a methyl group from S-adenosyl methionine. You can see that the methionine intensity decreases as the SAM methyl group intensity increases, and subsequently we get the growth of methyl peaks that are associated with the DNA. In this particular case, this is a small 10-base pair DNA, and we have methyl groups both at the top and the bottom when we add the methyltransferase DCM. And you can see that these two methyl groups are going to emerge with the same kinetics. How do we know when the reaction is complete? Well, we use the restriction endonuclease trick. We add restriction endonucleases, and if the DNA is not cleaved, that means that it's protected via the methyl groups, and one can simply read out the reaction time course with gels. So that we can produce the DNA that we want, we've generated a library of methyltransferases to do that. Some of them, again, place the methyl groups at the 5th position on C bases, some at the 6th position on A bases. This is a number of the enzymes that we have generated. They can form or generate methyl groups at either 2 positions or 1 position on the DNA. And we simply did these experiments to verify that the enzymes worked, and also that we had enough resolution so that we could add several labels at once to be able to look at multiple sites in our DNA. So now what we want to do is look at very large pieces of DNA, in particular, in this case, 153 base pair Widem DNA. This is going to be the DNA that ultimately will wrap the nucleosome core particle. What I show here is simply a one-dimensional NMR spectrum of a protonated version of the DNA or of the deuterated version of the DNA in either pink or black, respectively. And we use protonated or deuterated samples depending on the application. What I show here in panel B is a carbon, a proton HMQC spectrum, which of course is methyl trosified. This is on a 153 base pair sample, 100 micromolar concentration, 25 degrees, 40 minutes. And you can see that we get very nice peaks associated with the methyl groups of this large DNA. And superimposed on the spectrum, I have these single colored contours, which are the regions corresponding to very small pieces of DNA, roughly 10 residues, that contain the methyl labeling that we use to assign the black peaks to specific sites in the DNA. Now, there is a question about whether deuteration should be used. In the interest of time, I'll just summarize that by means of the following spectrum, the inset to panel B, where I show a carbon proton HMQC. And what I've done is I've taken traces in the proton dimension through the 5 different peaks that correspond to the 5 different methyl groups. The blue traces correspond to the spectrum, which is deuterated, of the deuterated DNA, and the red spectra of the protonated DNA. And you can see in the context of 5-methyl-Cs, there's a very significant advantage associated with deuteration, an order of magnitude increase in sensitivity. By contrast, for the 6-methyl-As, there's roughly only a 20% increase in sensitivity. So that if you had a system where you're only looking at methyls attached to A, you might not deuterate it. If you're looking at a combination of methyls both at A and C, you would deuterate. So it really depends on the application. And we've looked at now a number of studies. This is preliminary work focusing on the nucleosome core particle, where we've labeled both the protein component and the DNA component. So here's the structure of the nucleosome core particle, and what we've done is we've labeled H2B, histone 2B, that's the red protein with ILVs, and the blue dots here correspond to the methyl groups, which are decorating the DNA. We can record carbon-proton spectra, and by virtue of the fact that the chemical shifts are well resolved between the black DNA peaks and the red protein peaks, we have the opportunity to be able to do measurements simultaneously on both the protein and the DNA components. Now, we've looked at experiments that are fairly demanding in terms of sensitivity. We're interested, for example, in measuring picosecond, nanosecond timescale dynamics. And we use an approach that was developed many years ago for applications involving proteins, where we generate transverse protein, proton magnetization. It can be, of course, protein or DNA, methyl magnetization. And then by virtue of the fact that the methyl transitions are either fast relaxing or slow relaxing, we allow the system to relax for a period, which we call T-relax. That builds up a situation where we have different amounts of obviously the fast and slowly relaxing components, which we then exploit to create a triple quantum proton magnetization. And we measure the buildup of this triple quantum magnetization and fit it to an expression which takes into account a proton-proton dipole cross-correlated spin relaxation to be able to extract out the product of S squared tau C. And what I do here in panel C is I just plot out the S squared tau C values in black for the methyl groups associated with DNA, and then in red for the methyl groups of isoleucine, leucine, and valine that are associated with histone 2B. So you can see that there's a variation in the dynamics, and we can extract out order parameters. We can separate the S squareds from the tau Cs to say something about the picosecond, nanosecond timescale dynamics. We can also look at various biological questions. For example, we can ask, how does methylation affect the structural dynamics of the nucleosome, both from the perspective of the protein and the DNA? And we've just begun these sorts of questions. Or answers to these questions. It turns out that one common form of methylation is at the level of CpG steps, This is the most common form of methylation in mammalian systems and is involved in transcription regulation. And so what we've done is we've taken DNA where every one of the CpG steps in the DNA is going to be methylated at the C5 position of the C base. And that's shown here by these blue dots. And then we've recorded carbon-proton spectra focusing on the H2B methyl groups. And we've asked, are there structural changes in the H2B protein component? So here we have a superposition in blue of methylated DNA, the NCP derived from that, versus a wild-type NCP in red. And you can see there's absolutely no changes in chemical shifts. So structurally, there are no differences. Dynamically, there are also no differences as well. We can carry out this triple quantum experiment, and when we plot the S squared tau C values, both for the methylated sample and for the non-methylated sample, you can see that there's a very nice linear correlation. And the fact that we get data which is so reproducible, again, argues that we really have, enough sensitivity to be able to quantitatively analyze these sorts of systems. So that's our work that we've done to this point with the DNA studies. We're excited about carrying these much further, about looking at larger systems that are comprised of several nucleosome core particles and being able to ask important questions that relate to how dynamics controls function in these systems. I'd like to now talk about seeing the invisible. Now, typically when I give a lecture about seeing the invisible, I refer to conformationally excited states of biomolecules that are sparsely populated and transiently formed. I want to talk about a little bit of a different type of invisible today. I want to talk about the ground states of protein molecules. Now, you think about the ground states of protein molecules, we record spectra of these all the time, but what happens if you were looking at a protein which happened to be, say, intrinsically disordered, happened to be, say, an IDP, and you had to study the protein at pH 7. So then the backbone amide protons would become invisible to standard traditional sorts of NMR experiments. Well, in fact, that situation is more common than you might think. In certain applications, we have to study IDPs at pH neutrality. And the reason is because we want to look at interactions between several IDPs and the physical chemistry of phase separation. And phase separation in some systems only occurs at pH 7. I illustrate that here in the context of a pair of IDPs, caprin 1 and phosphorylated FMRP. And you can see that at pH 5.5, these are fluorescence imaging experiments. There are no blobs that are formed. Phase separation has not occurred. But as we go to pHs close to neutrality and around neutrality, you can see these red blobs. These red blobs indicate that there is phase separation. So we really need to look at higher pHs. And of course, we and others have developed experiments over the years whereby one can do that. One experiment involves starting magnetization from H-alpha, transferring it throughout the backbone, perhaps throughout the side chain, and then back to H-alpha for detection. Of course, H-alphas are not going to be influenced via the very rapid amide proton hydrogen exchange. And we can record backbone chemical shifts in a variety of different ways that allows us to get the assignments of the backbone for these intrinsically disordered proteins. But a problem emerges if we want to get the backbone amide protons, because those chemical shifts are notably absent from the spectrum. And so what we've done is as follows. We've carried out an H-alpha to C-alpha to carbonyl to nitrogen experiment, a very simple experiment. But when the magnetization is on nitrogen, we're going to do something a little bit special that is going to allow us to record indirectly the backbone amide proton chemical shifts. So it's a typical HCACON experiment, but when the magnetization is on nitrogen, we're going to record indirectly the proton chemical shifts. Let me just remind you of some of the mechanics of the experiment. These are things that we all know, but if we focus on a nitrogen proton two-spin system, for example, so we have a backbone amide nitrogen and an amide proton. If we don't decouple the proton and look at the nitrogen spectrum, of course, we end up with a doublet that is associated with the proton either up or down. From the proton side of things, I indicate this star to indicate the position of the proton resonance frequency. Now, if we apply a weak B1 field, say of a few hundred hertz, at the resonance position of the proton, then we will collapse this doublet structure to give rise to a singlet. And if we vary the position of this weak B1 field, say it's in chemical shift space, a position delta away from the star, which is the resonance position of the amide proton, then one can control the effective splitting in a manner that is well known, and this is sort of the zero order equation that describes the situation. So we can decrease the effective coupling from varying the position and the strength of the B1 field. Another brief reminder, again, is the fact that we're dealing with backbone amides, which are exchanging with water. And in the absence of exchange, so I denote the exchange by this factor alpha, which is just the ratio of the exchange rate, K exchange, to the chemical shift difference in radians per second between the two exchanging states. And the two exchanging states in this particular case, again, we're looking at nitrogen magnetization, are nitrogen associated with a proton in the alpha state and with a proton in the beta state. So we have a chemical shift difference in Hertz of roughly 100. And as a function of increasing exchange rate, then the lines broaden and ultimately coalesce via a self-decoupling. So this is the simple-minded picture of chemical exchange. And so we're going to take advantage of those two features in the basic experiment. So remember, we're beginning with magnetization from H alpha to C alpha to CO to nitrogen. When we have the magnetization on nitrogen, we then invoke the following element that is shown here. It's a spin echo where we apply simultaneous proton and nitrogen pulses in the middle of this echo. So at a time tau of a 2 tau delay period, we apply simultaneous proton and nitrogen pulses. If we begin with magnetization that is NY, say, and let's choose for the purposes of argument that this 2 tau delay is chosen to be 1/2J, where J is the nominal proton-nitrogen coupling, which is, say, 94 Hertz. So if 2 tau is 1/2J, and in the absence of any exchange effects or B1 fields that we might apply, this nitrogen in-phase magnetization is going to become completely antiphase by the end of the 2 tau period, and we're going to not observe that magnetization. Okay, this experiment is geared to observing in-phase NY magnetization at the end of this 2 tau period. Now, what we're going to do is we're going to apply this weak B1 field, as I said, and we're going to carry out a series of experiments whereby we vary the distance, the spacing, between the application of the B1 field and the position of the invisible proton line. And as the position varies, as delta varies, the effective J coupling will vary. The coupling will become smaller, and as a consequence, during this 2 tau period, which is set for a coupling of 94 Hertz, if the effective coupling is smaller, there will be some residual nitrogen Y magnetization at the end of this 2 tau period. We plot that residual magnetization here as a function of the position of the B1 field, and where we get the maximum intensity, that's where the position star is. That is to say, that's where the resonance position of the proton is located. So the experiment in the context of when we have an exchanging system with water is exactly the same as what I indicated, except now the exchange with water effectively, again, attenuates the scalar coupling. And so even in the absence of a B1 field, at the end of a 2 tau period, which is set to this 1/2J, where J is 94 Hertz, we're going to have some residual magnetization. And that's shown here as a function of K exchange. If K exchange is 0, there would be no residual magnetization because it would be all antiphase at this period. But if J, or if K exchange is not 0, we end up with some residual magnetization, which increases as K exchange becomes larger. Eventually, K exchange will become so large that it will effectively self-decouple, and then we'll end up with a maximal signal independent on the position of the weak B1 field that is used to probe for the chemical shift of the proton, the invisible proton. And at that point, there's no information whatsoever from this experiment. Of course, where that occurs depends on the experimental condition. So if we use a B1 field of roughly 400 Hertz, if 2 tau is set to 1/2J, where J is again about 94 Hertz, then K exchange values up to about 1500 still can be used. But for larger than that, we would get flat lines. In that case, we have to increase the B1 field, we have to increase 2 tau, and then we can observe these profiles that allow us, again, to place the chemical shift of the invisible state. I should mention that these sorts of ideas are prevalent in the literature. Jeffrey and Franz have used these kind of ideas to measure K-exchange in very fast exchanging systems. We're not interested in K-exchange per se here. We get that information sort of de facto, but we're interested again in looking at position of invisible peaks. Other people have used the idea that one can reduce dimensionality in high dimensional experiments through the application of a B1 field that changes the line shape of a directly detected nucleus, and in so doing get information about chemical shifts. Those approaches would not work very well here because we have very broad lines, very broad nitrogen lines, which are due to exchange. And so we've done this sort of experiment, which is based on intensities as a function of position of the weak B1 field. And on my final slide, let me just show that, in fact, we are able to see the invisible. I show a proton-nitrogen correlation map of Capron-1, this intrinsically disordered region of Capron-1, actually, of about 100 residues. This is at pH 7.4, 30 degrees. This spectrum is of very poor quality, and in fact, we don't see the majority of our glycines, which would be here. But by doing the experiment that I just described, these glycines all of a sudden emerge, and we can get their chemical shifts simply by the position of the maxima in these profiles. Very interestingly, there are 4 glycines in this region here. A couple are observable, but some are actually not observable in the traditional nitrogen proton correlation map. By seeing the invisible, we can establish that these backbone amide protons are actually shifted upfield. And it turns out that these amide protons are associated with glycines that are 2 residues downstream, if you like, from aromatic moieties. And so we think that the glycine backbone amides are pointing into the plane of the aromatics. And if that's the case, this would imply that despite the fact that we have an intrinsically disordered region of a protein, there nevertheless is residual structure. And of course, the question now emerges is whether this residual structure is going to be important for phase separation. And we're exploring that as well. So on that point, I will stop. I want to acknowledge the people that have done the work. Gilly and Al really did a tremendous job with the NCP story and in bringing methyl groups and methyltrosy to nucleic acids. Pramod visits us occasionally and really inspires us. And the seeing the invisible experiment was really due to him, with assistance from Enrico and Leo and Taehoon. I thank them, and I thank you for your attention. And I think now I'm supposed to ask questions that you all have. The first question is, how do you decide the DNA sequence in the NCP? Is it human DNA or randomly decided? It is not human DNA, and it is not randomly decided. Initially, we have been working on DNA that is the Widem 601 sequence. So the Widem 601 sequence is a positioning sequence. So it really positions the DNA with respect to the histones, and it forms a very stable complex. And you would want to have a very stable complex initially when you start experiments, because you want to get the best spectra that you can. Now, our DNA is generated by expressing a plasmid. It's a high copy number plasmid, something like 30 copies of the 150 base pair sequence are present on this plasmid. And so since we had that plasmid around from our previous studies, that is where we started. And of course, that limits where we can place methyl groups, because there are methyltransferases. The methyltransferases are very specific. They recognize sequences in a very specific manner. Of course, if you have 153 base pairs, there's going to be lots of sequences that can be recognized because there are lots of methyltransferases. And we've generated, 6 or 7 that can be used. So there's a little bit of prior history into why we decided to work with the system that we did. Having said that, now we want to work with other perhaps more relevant DNA, like human DNAs, and see in systems where there's more dynamics, how it is that methylation is affected there. Presumably, it will have a bigger effect because the DNA sequences will be less stable. We also want to look at how mutations at the level of the histones will affect less stable nucleosome structures. I mean, nucleosomes are not static entities. There's something like 35,000 entries in the PDB. These are static entities that really don't explain how it is that, you know, readers of epigenetic marks can invoke such, you know, significant changes in nucleosome structure. You've got to be able to unwind the DNA to be able to act upon it. So these have to be very plastic, and we think that NMR is well poised to get out atomistic descriptions of the level of the of what the plasticity means in terms of structure. There was a question about, do we linearize plasmid DNA? And the answer is yes, we have to chop the plasmid up into its DNA components. The questions are going by quickly, and there were questions, no answered questions, it tells me. How large an IDP can be studied using the method you spoke about? That's an interesting question. I will say that that's a two-pronged question, or at least there's two-pronged answers. One of them is size in terms of molecular weight, and the other one is size in terms of resolution. So in terms of molecular weight, I think we can go quite large. The reason that I say that is because I didn't show it today, but we've done these sorts of experiments on phase-separated proteins. And there, of course, it's an incredibly viscous environment. The diffusion of proteins in a phase-separated milieu is sort of akin to the diffusion of an E. coli cell. So these things diffuse very slowly. Certainly there are internal dynamics, which is critically important for getting high-resolution spectra, but the overall dynamics is much slower than would be the case for a normal IDP under conditions that are not phase separated. So we think that we can go to significantly larger IDPs from a perspective of relaxation. From a perspective of resolution, the experiments were recorded at 600. There's now, of course, much higher fields. That will help in terms of resolution. One can also invoke higher dimensionality experiments, which should also help. So I think the prospect is good for going to larger ADPs. Did you label both methyl groups for leucine and valine? The answer is sort of. We use the labeling approach that Vitaly Tugarinov, when he was in the lab, proposed, which is that one of the two methyls is C13H3 labeled, the other is C12D3. That's important to minimize a crosstalk between the methyl groups. But at the same time, we also do not stereospecifically label the methyls. In this particular case, we have good enough resolution because the histones are fairly small, so that at any given time, either the pro-R or pro-S methyl is labeled, but we have both components labeled at 50%. What are the time requirements for these experiments? So I'm not sure what experiments you're talking about, so I'll give an answer that refers to all of the experiments. The answer to that is for the methyltrosy-based experiments, we can get a spectrum in 15 or 20 minutes on a 100 micromolar sample. You know, and that's really, I think, two things there. Methyl groups are sensitive, and Bruker cryoprobes are sensitive as well. When we want to do longer experiments or more quantitative experiments, like the relaxation dispersion experiments, like experiments where we measure S squared tau C values, there we typically record experiments for a few days, 3 days, I would say, and we do that whether we're dealing with DNA or applications to proteins. Remember, these are hundreds of kilodaltons in molecular weight, and you're only as good in some respects as you know, your least sensitive peaks. So we want to be able to see that we can measure those as well. There's a question, are methyltransferases commercially available? How many of them did we use? Are there methyltransferases for RNA? Methyltransferases are commercially available. We thought that we would go that route and simply buy the enzymes. One can do that. But it gets to be very expensive, and so we produce the enzymes ourselves. We bought the DNA, and we developed the protocols for expression of the methyltransferases that we then need for labeling. And we're happy to share those protocols or the methyltransferases with anybody who wants to. both if you want to collaborate or if you just want things to do yourself.
Louis, maybe
Yeah.
in the interest of time, we can you can answer the remaining questions at the end of the seminar. How about that?
That's perfect. Perfect. Thank you.
To all the attendees, your questions will be stored somehow in the Q&A, and both speakers will be glad to answer your questions afterwards. Right? Sorry for interrupting.
Oh, fine.
So, all right. In the interest of time, I would like to just get started introducing our second speaker, Daniella Goldfarb, to talk about EPR. So Daniella's lab at Weizmann Institute of Science has pioneered the the use of spin labels such as gadolinium and other paramagnetic metal ions to do near EPR distance measurements. And because of the high sensitivity of gadolinium and compatibility with cells, they're now bringing these technologies to very interesting and important biological questions on IDPs and protein conformational changes. So Daniella.
Hi everybody there. It's my first big Zoom seminar, so I hope it's going to work okay just speaking to the computer and a couple of faces. I'd like to thank the organizers for putting it together, and so I'll start. Today I'd like to talk not for gadolinium on a change, but mostly on manganese, and to talk about how we can use manganese distance measurements to learn something about HSP90. And this is a project that we've been working on for the last couple of years. So HSP90 is really a hub protein, a very important one. It's a molecular chaperone, and it's conserved from bacteria to humans. It interacts with several hundreds of other proteins that are referred to as clients, and it facilitates their folding and maturation. And these clients are usually involved in many important cellular functions and pathways. So it turns out that HSP90 is really a very important protein. It functions through, firstly, it's very, very flexible, and it functions, it undergoes several conformational changes and gets the energy from ATP hydrolysis. And its flexibility is really a key of its function, and it can be regulated by different co-chaperones. So they can change the rate of ATP hydrolysis and stabilize several different conformations. And like this, I guess it gets specificity to work on so many different proteins. HSP90 is also a drug target where you can, inhibitors targeting the ATP binding site can stop it from functioning. So we were interested in its dynamics. So there's it's a big protein. It's a dimer of one. Each monomer is 90 kilodalton, and this is a crystal structure with the ATP analog AMP-PNP. And this is with a co-chaperone. I stripped it, but so you can see the protein. It has 3 domains. Here is the C-terminal domain that is really in charge of the dimerization, and it has to be dimerized to work. The N-terminal domain is the one that binds the ATP. And then there's the middle domain that is important for the ATP hydrolysis and the binding of clients. So this is in the crystal structure, and there's been a lot of work currently with also electron microscopy that gets structured. There was a lot of NMR, mostly on the different, on the isolated domains, but recently there's also beautiful work on complexes. And a lot of fret work. Fret work was done mostly in solution. And the picture that comes out in terms of conformational changes throughout the ATP cycle is at the beginning we have the apostate, and you have these 2 parts of the dimer, and here there's this very flexible linker, and here is the dimerizing part. So this is the C, M, and N domains. Now, upon binding ATP, there is an internal conformational changes that goes inside here, and then it closes. It goes to the closing state, and this is the functional state. Now, from Fred, they figure that also in this state there is an equilibrium between the open and closed. Then one goes to what we call the high energy state, and this is where the gamma phosphate is cleaved, but it's still on the protein. Then it leaves, it goes into another state that is still closed with the ADP before the ADP is released. And here, there was some debate in the literature from electron microscopy from a different type of so we worked on the yeast, but on another one, they saw a compact state. Some others say that with ADP it's just open. So this bit was a bit controversial. And then once the ADP is released, it goes back to the cycle. So this is without the chaperones. And we were wondering whether we could really track something here, 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 and here using their distance measurements. So how do you trap the different states of the hydrolysis? So the hydrolysis is pretty slow. So the pre-hydrolysis state you can trap with, so in all of these experiments, you have to have magnesium. Without magnesium, it doesn't hydrolyze, so magnesium is always there. So you can either add magnesium ATP and freeze very fast. Anyway, we do all the measurements under freezing, and then it's still in the pre-hydrolysis state, or you use the non-hydrolyzable analog that is given here. Yeah? The high energy state is usually obtained by adding ATP in presence of vanadate. You wait a while and it's trapped there because it doesn't release the inorganic PI. And post-hydrolysis, it's either you add ATP, you wait 2 hours and the reaction is finished, or you add ATP. So these are the 3 states that we looked, and we tried to see whether we can follow some of these conformational changes. So here is again the protein, and these are all the different sites. So we have 2 sites in each domain. These are in the C. We have 2 in the M and 2 in the N. Here is the position of the nucleotide where it binds. And we started by using gadolinium spin labeling because we had the idea that once we understand this protein in vitro, maybe we can go and work in the cell. So let me tell you that it's going to take a long while till we go into the cell because it's pretty complicated to work on it outside. So that's why we started initially with gadolinium. This is the label that goes on cysteine residues. And we also tried nitroxide. The usual MTSL did not work very, very well, so we tried this proxy. So the experiment, for those of you maybe forgot or didn't know, is very simple. Basically, we measure the distance between the 2 spin labels. This is just measuring the dipolar interaction, and we all know that it is proportional to the distance between the 2 paramagnetic centers. And then there is a orientational function. We work in frozen solution. All these angles have equal probability, so this we know how to handle. And what we're looking after is for this distance distribution. So basically what we measure is the dipolar frequency, and this is further translated into distance. So it's a simple echo experiment. You can think about it as even a pump probe. I'm not going to go. This is just collecting a refocused echo, and this is just another bypass that is supposed to cause dephasing and dephasing, so the echo is not fully refocused. So once you measure the echo intensity as a function of the time of this pulse, you get these oscillations, you get rid of this background, you get these oscillations, and then you can get the distance distribution. Now, you can imagine that when you work with a protein like HSP90 that is very flexible, you will not get such very narrow lines. You freeze all these different conformations and you get a big, now it depends how broad is your distance distribution. Now your spin label also has a tether, so the spin label is connected to the cysteine, and then the spin label itself can add some weights to this distance distribution. So you would like to work with more rigid spin labels, and this gadolinium label that I showed you before is pretty rigid. So we started with the C terminal that is supposed to be dimerized in all states. So this was mostly a test. So we labeled in this position, okay, here, and we checked we did this deer experiment with the APO, with AMP, PMP, and ADP, and you see this is the distance distribution that one gets, and this is what you would expect if you graph this gadolinium label on the structure. You have all these distances, and it gives you this broad distance distribution, and you see that it agrees very well. So this tells us that the C domain is dimerized in all states, and also we get pretty close to what is predicted, and then we just did another one, and it behaved the same. So this is just another mutant. These are the distance distribution, and you see it agrees pretty well. So we were really happy. So now we have to go and measure the N domain and the M domain, which are the more interesting one, because that's where the action is. So there, the labeling with gadolinium did not work very well. Either we got very low labeling efficiency or protein precipitate. It didn't really work. So we said, okay, so we need to compromise and go to the nitroxide. And this is what we got with the nitroxide. And you see it's terribly broad. This is the predicted one. There's no information here. So we went to another mutant and was just as bad. So we're stuck. But then we thought, okay, we can do something else. This protein, as we said, it has an ATPase site, and this ATPase site has to have manganese. So there is a very well-defined position for the manganese to sit as it's coordinated to the ATP. So let's focus on the crystal structure. And here is the nucleotide, and here is the manganese, is the magnesium. Now, in EPR, it has been for many years, people have used manganese as a paramagnetic probe for magnesium. So we said, okay, let's change the magnesium with manganese and then try to measure the distances between the two manganese. Maybe we will be able to see something. So first thing one has to do, we had to check that manganese is still active. So it does. And it gives very similar ATP rates as magnesium. And now, if we do this, what do we gain? Because here we really have a spin label that is a point. It's just an atom. So we don't. So if we can measure it, it really tells us the dynamics of the protein without all these extras from the spin label. And also, the spin label sometimes can perturb the structure. So this is more naive. But there's some complications. So in principle, the binding constant of manganese, ATP or ADP to the protein is not very large. And there are two equilibrium equation one has to worry about. It's the binding of the manganese to the nucleotide itself and of the manganese nucleotide to HSP90. Luckily, these are in the literature. So one can calculate for particular concentrations that we can afford, such that we have a large amount of dimeric HSP90, where you have manganese in both monomers, and this is what came out. So for instance, the binding of ATP is a bit weaker than ADP. So here we have only 25 of the dimers having both manganese in both monomers, and for ADP we have 64. So this means that we should have larger modulation depths here than here, because here we're gonna have many background. It's manganese ADP that it's not bound, and we have also molecule dimers that have only one. Nonetheless, we decided we should give it a try. So we really wanted to know in what state we are. Are we really before hydrolysis, after hydrolysis? And here one can use ENDOR. So ENDOR is just a method where you can measure the NMR spectra of the coupled nuclei. So if you do ENDOR, you can measure the phosphorus hyperfine interaction. So these are the phosphorus signals. And what I just want to show you here, that the line shape is very different if there is if you are pre-hydrolysis, high energy state, or post-hydrolysis. So I guess maybe for you it all looks the same, but if you look carefully here, this is this shoulder, and this shoulder disappears upon hydrolysis. So we see that if we just freeze it very fast, or we use AMP-PNP, we still have these shoulders. For the high energy state, it looks really like ADP, and this is for, if you let it work and leave the sample for 2 hours, everything that was converted to ADP. And here, so from the ENDOR, we know that we are okay and in what stage we are. So this is another nice benefit of the manganese. Okay, so now we go to the distance measurements, and this is what we got from the pre-hydrolysis state. And these are just triplicate, and it's different. It's 3 different preparations because the modulation depth is low, and we had to make sure that these are really real. So these are the pre-hydrolysis. So you see mainly a peak at around 4. And then for the post-hydrolysis and for the high energy state that we know from ENDOR are very similar in terms of the region around the manganese, we see that the distance distribution is a bit broader and shifted. Now, if you take from the crystal structure, this is what you predict. So there were two different structures, one from electron microscopy, and we are a bit short. And if you look, you see here there's also a difference in the modulation depth between the post and the pre, and this is what was predicted from the binding constant. The difference is a factor about 2.8, 2.6 was predicted. So we felt that pretty good with this. So what did we learn at this stage? So we learned first that this is, in all cases, the C domain is dimerized, and these are the distance that we measured. For the ATP, we found a closed state that is relatively narrow, and it has this distance. And now we were able to trap the ADP closed state that is different from the ATP. It's a bit closer and a bit wider. So we were able to differentiate these 2 different closed states. Now, whether there is an open state, we don't know, we didn't see it. So maybe the distance was just too large for us to detect it, and but we didn't see it. So if we go back to this cycle, so we were able to experimentally see the ATP bound state and the ADP bound state, and we also saw the high energy state that is pretty similar to the ADP state. So this was this part, and now I just want to go to some experiments that are more preliminary that we are doing today. Now, so up to now we looked at the opening and closing. So this, we had one manganese on one monomer and on the other, and so we were sensitive to the motion of these 2 monomer, one with respect to the other. But there could be that there is some motion that goes between the N and the M domains, like something like doing this. So we really wanted to measure also intramolecular distances and not only inter. So now if want to do it with nitroxide or with gadolinium, we're going to end up with 4 different distances, because we have one distance that is intramolecular, but then we have 3 that are intermolecular. So we already saw that we had problems with the nitroxide with the width. So this would have been hopeless. But then we said, okay, let's try and do manganese nitroxide. So we can use the manganese in one monomer and measure its distance to the nitroxide in the same monomer or to nitroxide in the other one. So in this case, one has also 4 different distances. Yeah, so we have, these are the intra one, and then you have the inter one. But we can spectroscopically select such that we don't see nitroxide, and we don't see manganese, so we are left only with 2. So this is the exercise now that we wanted to do. I just want to mention that all these experiments are done on a home-built high-field spectrometer, and to see manganese, it's much easier at the high field because it has 0 field splitting, and just like quadrupolar nuclei, the higher the field, the narrower the signal, and the more sensitivity you have. So here we have preliminary results. So here, this is one of these different nitroxide labeled HSP90 I showed you before, and this is with AMP, PNP, and ADP. Here we don't see much differences, but what you predict from the structure, the two manganese nitroxide distances should be the same. So we see here a closed one, but we see also this large one, and I think that this one comes from the open one. So we were able to see through the manganese nitroxide also the open one. So you also have HSP90 that has the nucleotide bind, because we see only the ones that have manganese. Those that don't have manganese we don't see, and we see that even with the nucleotide bind, they still have an open structure. So now the question is, what happens with, so this is this open one. So the question now, what happens with the co-chaperone? This is the crystal structure I've shown you before, and it has the chaperone here, and it could be that the chaperone is the one that helps it to go into the closed state. So we said, okay, let's try do it with and without chaperone. These are all preliminary results. We have to check it on more mutants, but I think it's relatively promising. And you see that without this co-chaperone, we see the open one, and without it, it's really disappearing. These are just codes that tell you what is the reliability of the distances. So when you have it green, both the distance and the distribution are reliable, and you go into the yellow, the width is reliable and the place, and when you go into the orange, the mean distance is reliable, but the shape is not. So in any case, we see the reduction here, and then we said, okay, so let's see what happens to the manganese, and here we just did the manganese measurements with this co-chaperone, and you see that also here we see a closure, and it goes, and you remember before our distances were a bit too long with respect to the crystal structure, but now it really comes in the right place. So I think this is all I had today. So all to say, this will be continued and confirmed. So the last 3 things need to check on more mutants, but I think it's promising, and it's really a way to go around this nitroxide spin labels that did not work for us for this very flexible protein. Now I want to thank the people. So most of this work was done by a very talented postdoc in my group, Angeliki, and she works just as well as she smiles. And Akiva Feintooth that helps with the spectrometer, and these are all other people that helped a bit with the biology. And this is the group before Corona times. And I think with this, I thank you. And I'm happy to take questions. I have to find them somewhere. I don't know how I find the questions now.
It's in the Q&A.
Here, Q&A. Now it shows up. Okay. Daniela, do you have any idea what? I don't know. We couldn't get any, it didn't work at all. Did not work very well. And what was surprising for me, we chose, we chose the labeling position according to fretwork, where they put fret labels, these big dies. So MTSL did not work, the proxy worked, but still we got, I don't know, the data were just very, very broad. Why are there such big differences in the, this is not for me, the TROSY. Do you ever get anomaly short distances if you are exciting the, no, because you see the experiment, when you do the deer experiment, your pampas is just making a difference of 1 quanta in the spin system. So it doesn't matter if you hit a 5/2 transition or a 3/2 transition, because you would just flip by 1 quanta. So then you just see one dipolar frequency. There are some, maybe some second order effects, but it would have been nice if we could excite more. So I went to Lucio Friedman and I asked him, how can I do multiple quantums so then I can see longer distances? And he told me, just give very, very strong pulses, but I don't have. How did you decide which system to use for labeling? So we followed the fretwork. They did many different mutations and put these fluorescent dyes, and we looked at their results and looked for distances that we thought that we could access. Lauren would like to answer this live. How do you do this?
I'm not sure how it appears on the other side. I was just trying to moderate the questions so not all of them fill up the queue. But you can also push answer live and then it will display correctly, probably.
Okay, so I think I don't have any more questions, right?
Could I ask one?
Yeah, sure.
You mentioned this system where you had 2 manganese and 2 nitroxides.
Yes.
And then you selected manganese to nitroxide,
Yeah.
but you suppress the manganese to manganese. And in principle, in NMR, that would be that you can suppress homonuclear coupling, but only see the heteronuclear coupling, which is difficult in NMR. So how do you manage that technology?
Because it depends where I sit on the spectrum, and also for instance, for manganese, I need different power than for nitroxide, so I can play with the power, so it's not so effective for the pampas. Yeah, so usually I observe the manganese because it has a fast relaxation, and then I flip the nitroxide. So then I don't care about the T1 of the nitroxide, can be slow. And then, so I can play with the position in the spectrum. So I can do manganese if I go out of the nitroxide spectrum. I can do nitroxide if I'm giving pulses that are more appropriate for spin half than 5/2. And also the manganese has very shallow modulation depth. So usually I have to go out to see it. So this is spectroscopic selection. For Daniela. Okay. can you talk about the background subtraction? What happens if you get it wrong? Where does the background come from? How do you decide what to subtract? Okay, so the background comes because you have, so when you have all your paramagnetic molecules in the solution, then you have interaction within the molecule. But because this electron-electron dipolar interaction are long range, then you have contributions from different molecules in the solution. Yeah, and these have all, it depends on the concentration, and this can have, you know, all distances, and this turns out to be just an exponential decay if you have a homogeneous solution. So usually you measure long enough after your modulations have died, and you can estimate your background decay, and then there are methods to check how much of an error is introduced in there. And I can, for instance, if I go back in here, wait, how do I go back? Yeah, not here. One more. Yeah, so you see here, this width here, this comes from background uncertainty, but you see that there is a peak. Yeah, so if it is 5.2 or 5.4, doesn't matter, but you have a peak, and this comes from the background subtraction. But this background, if you don't have long enough traces, is an issue. Okay. There was a question about AWG somewhere. Have you tried using, everything is with AWG, all these measurements. So AWG means that we use chirp pulses to excite, for the pump pulse, so we excite larger parts of the spectrum. Do you think you are seeing an equilibrium between the open and closed state which shifted by the chaperone, and you can quantify? Yeah, I think I see it. I'm not sure if I can quantify it. Maybe, I don't know. This is for the future. That's what we think. Since both gadolinium at sulfate board, how do you manage to introduce different labels on a single protein? No, we did not. The gadolinium was on one mutant, and the nitroxide on others. We did not have both gadolinium and nitroxide. We had both manganese and nitroxide. Lauren, you want to answer? Okay, so we can you use cysteine present in the protein without requirement of mutation? Yes. If you have cysteines and they are not functional, then you can use them, and it saves you a lot of time and effort.
There's another question, Daniella, about magnesium ion for activity.
Yes, so you have to have magnesium. Unfortunately, magnesium is not paramagnetic, and I know that also NMR is not easy on magnesium. So that's why we use manganese instead.
Okay. All right.
But it is as active with manganese as it is with magnesium?
Yeah. It's just 20 minutes, but you know, all the spin labels, everything was active. So I don't know active, but they did ATP hydrolysis. You know, I don't know if they act as chaperones and folding, but at least they hydrolyze the ATP.
Okay, I think Daniella has answered most questions. Since it's 12 past the hour, I think today we're at a close, and thank you to all the attendees for the questions, for coming to this seminar series. And next Thursday we resume at this time. Again, if you have further questions, please continue to contact Luis and Daniella. And I think we have a poll, right, Marcus? Do you want to say anything about the poll?
Yeah, I mean, as May said, so we have a very little poll just to find out if there's a severe overlap with any other webinar. So please vote on it, and we might consider then this in the future meetings. Thank you.
Well, we hope we don't change this time. We will try to keep this time, but if you have serious conflict, please do also let us know. So, all right, with this, we'll say goodbye.
Yeah, please, one more thing. We intend to make a website which will be called emergingmr.org. emergingmr.org, where you can then look also whether there are any further informations regarding this. Also maybe the time for meeting the speakers and so on, if that is going to happen. So just watch out for that, emergingmr for magneticresonance.org. It may not be online tomorrow, but it will come online hopefully within a week. And next time we also hope that we can have more than 500 participants. We were above 500, so the success was overwhelming, and we also hope that this is going to happen. So please tell all your friends who could not log in that next time they will be able to join.
And today's two talks will be on the YouTube on the ICMRBS website. Okay. All right. Thank you.
Okay. Bye.